that could not be done.
Yes, sir. Yes, sir.
And therefore, since it is mandatory to have presentation of fellow awardees and all awardees, as has already been decided by the executive body of SPR, therefore these presentations have been kept.
Okay.
Of course, I fully understand that the number of audience and the pupil who will be joining the third part of the series may not be that much, because we were having short time to declare it, and secondly that we are in a position to dispatch all the awards, because no conference is scheduled in the near future within 1 or 2 months. So we thought that better the awards and mementos should reach to the awardees by post, and whenever the conference will be scheduled, they will be duly informed and they may come and join. So before we proceed further, let me give a very brief introduction, because today we are finalizing this lecture series for esteemed fellow awardees. The executive body of Society for Plant Research, which has been established 36 years back in 1988, and after a long journey of 36 years, this society and its publication Vegetos has now attained a global status, and it is very well established. But one point which I want to emphasize this evening, that being globally established is not our target. The target of the society is to strengthen the journal and the society further by collaboration, active collaboration, dedication, and support of all scientists who are associated to it. Honoring the contribution, the research findings of a scientist is very important, and the executive body has decided to recognize the contribution made by the scientific community, fraternity, and to recognize them duly by providing them a token of appreciation in the form of award and a memento. But getting award means you are closely associated, you are a part and parcel of the society and its journal. So after award, your journey starts for the society and for the journal from this moment. I am sure that we all will work together and will do everything possible for its more glorious journey the worldwide, its better establishment, more improvement in the journal, and more suggestions, creative suggestions for the improvement of society. I am fortunate to have very young group of scientists, because when we established the society in 1988 as a founder, Secretary General, we never had a dream, or we never thought that we will reach to this global status. Of course, that was our dream, but with the passage of time, we could grow systematically, and the society and its journal has now established itself very well. So, instead of going into details of it, I would request Dr. Anayatullah Siddiqui to kindly continue with his presentation, which will be followed by the presentation of Dr. Gopalakrishnan. Dr. Anayatullah Siddiqui, please. Your mic is mute, Dr. Anayatullah.
Yes, sir. Okay, sir. I am uploading this presentation.
Oh, in case you find any difficulty, our office can help you in your
Sir,
presentation.
already I have shared my presentation to the office. If he can
Yeah,
upload from that side.
Should it be done from our office or you will do?
Sir, I have shared. If he will do, then better.
Okay,
Because
okay. I'll ask my office to kindly share the presentation of Dr. Anayatullah Siddiqui.
Okay. Sir, can I start?
Yeah, sure. With the permission of President, you can start.
Okay, sir. Thank you very much for inviting me as a chairman, co-chairman, dignitaries, SPR fellows who have been selected for this award. First of all, I would like to show my deepest gratitude to the committee member who has been selected me as a SPR fellow, and I'm committed to do better for the society. Now I'm going to start my presentation. The topic of my presentation is seed priming as a strategic tool, potential, limitations, and future directions. Now next. In the present scenario, what content I will deliver today, the topic of content is what is seed priming, basic behind of this mechanism, type of seed priming, which is nowadays and in past have been done by the several scientists, potential and benefits of this priming techniques, limitations and challenges which are faced by nowadays, and future direction in brief I will give. And in the last, I will present conclusion. Next. I would like to start from the seed priming, what it is. Everybody knows it is not a new technique. It's a very old techniques. Since long back time, people are using. But after the advancement of the time, the concept and mechanism has been established. First of all, this priming was scientifically, concept-wise established by Heidegger and his co-worker in 1977. After that, even though he has given that term seed priming first of all, seed priming is a pre-sowing techniques that activates early germination process without actual emergence. Basically, it is between the game of hydration and dehydration process. And if I would like to define, seed priming is a technique by which we can invigorate the seed in a respective solution for a certain period of times. Next. Why seed priming is important? This is very important to understand why seed priming is important for us nowadays. By using these few headings, I would like to explain to increase the rapid germination. Germination of the seed is the foundation of any kinds of receiving the good yield of any crops. If out of all the seeds which has been sown over the field are unable to germinate, it means it is going to impact directly on the production by reducing the population. Therefore, we will use, people are using seed priming to increase the germination percentage up to optimum level. Second, to increase uniform seedling establishment, which is again a very important topic, because germination is one process, but if it is not healthy and not going to establish over there properly, then mortality rate will increase, and again the population of the field will reduce and ultimately will impact the production. Third one, to mitigate the response of stress. This is again very important things. First of all, once we sow the seed over the field, we know very well the first adverse conditions are going to face by seed in the soil. That may cause several kinds of stresses, maybe kind of water stress, you can say nutrient stress, you can say temperature stress, or even though nowadays it is very popular, several kind of stresses you can say will be faced by the seed. Next, to increase the seed quality. Quality in the sense how rapid mechanism of the breakdown of the endospermic material will start. Thus, it will increase the later process to get it germinate well over the field. Next, last one, to increase the yield with low input. Low input in the sense whatever chemicals we use, whatever agents as a seed priming we use to invigorate the seed or as a seed priming, which is very cheapest price, because the concentration and amount of chemicals are required in very low amount, and by which we can increase the yield. People are asking very frequently this question I am facing every time in conference, how this priming is going to impact directly to the yield. So in this regard, I would like to address over here, priming will not increase directly yield, but we know very well the growth of the plant is a sequential event. This is not a direct phase, it's a sequential event, and priming start their work since beginning from the germination, and every stage is depending on the previous stage. If previous stage of growth will be good, then definitely later stage will be better. So in that way, it will increase the yield. Next. If I talk about type of seed priming, we can categorize into two major categories. One is the traditional seed priming techniques, and nowadays, which is very popular, which is known as advanced seed priming techniques. One by one, I want to show you. First one is hydropriming. It means by using water, or sometimes people are using distilled water, even though, to invigorate the seed for a certain period of time, and their response appears during the seed germination. Next one is halopriming. Several chemicals are in this category. Basically, the purpose of halopriming is to increase the tolerant capacity against the salinity, which is again a very big problem in India. For that, sodium chloride, potassium nitrate, potassium chloride, likewise chemical people are using. Next one is osmopriming. By using PEG, the polyethylene glycol, mannitol, sorbitol, many chemicals that we use as a osmopriming, and it is very important. Again, solid matrix priming, this is the next one. It's basically organic carriers we use to make layers around the seed by using some chemicals, which is very popular. Exofolicate, vermiculite, and calcium silicate are few basic examples, and it helps in increasing supply continuous moisture to the seed after placing into the soil. Next one is hormonal priming. Is a very common. In this kind of priming, people are using different kinds of hormone, especially abscisic acid and auxin are very common. Next one is nutri-priming, in which people are using macro and micronutrient as per their requirement, what he want to supply along with the water to the seed, they are choosing. If I talk about advanced seed priming techniques, in which one of them is biopriming. In this process, we use different kinds of, you can say, bioagent like rhizobium, PSP, phosphate solubilizing bacteria, their strain, and KSP is a new, you can say, biopriming agent. Even though that KSP I am using and getting very good result for the yield purpose. Next one is nanopriming. This is a very latest techniques people are using because it is very good because it can precisely deliver the nutrient, respective nutrient, to the seed along with the water to get it benefit, in which silver, copper, titanium dioxide, zinc oxide, and many more are popular. As per their requirement, people are using and getting their benefits. Last two one is the magnetopriming and gas plasma activated water. These two are not yet established, but yes, under the research, it is very common. People are using magnetopriming seed exposed in magnetic field before planting over the field. Their mechanism yet to be established now. And last one is gas plasma activated water, which is rich in reactive oxygen, nitrogen species, and other compound. First of all, this gas plasma activated water concept has been patented by a scientist known as August Jones, Mary Peeman in 2015, and nowadays in some research paper, people are using this technique also to get their benefits. Next. Here, I want to show you the background of seed priming, how it works basically. In right side, I have an image of seed, which is showing the internal process, how the entry of water can influence the gibberellic acid, as well as their, you can say, DNA, RNA, followed by activation of amylase activity, and that amylase will act on the endosperm to get it break, and thereafter energy will release and will participate as a source of energy into cell division. But what the significance of showing this image in relation to seed priming? Left side, one graph is there. That graph I have taken from my own research paper, which has been published in last 3-4 years back. And I want to show you triphasic process of seed priming, in which this black color line is showing about, you can say, unprimed, and this orange color is showing their primed seed process. Unprimed process will, once will start, may not be able to stop them, will continue. But in case of primed seed, we can go up to second level, second phase. There we can get it dehydrated, and that seed we can use later on as per our requirement over the field. What will be the benefit? At least 4 to 5 days early, seed can germinate if we use primed seed over the field. Next. Here, the seed priming is background. By using this graph, I want to show you if we use primed seed and unprimed seed, if we compare, we can see within, you can say, 30 hours, entire seeds are going to be germinate if primed seed we are using over the field. And if unprimed seed we use, we can see it goes up to 90 hours. It means directly 4 to 5 days benefit will be there if you go through the seed priming techniques. Not only early germination is possible, along with them, healthy seedling is also possible by using different kinds of priming agent. Next one. This is my latest work that I want to show over here. Nowadays, I'm focusing on nanoparticles based priming I'm using, and I use zinc oxide over here. And I established 3 things in this, you can say, work. First thing, I have focused on concentration and duration. These 2 different things is very important. If concentration and duration of the seed priming is not optimum, then we cannot get 100% benefit of seed priming. So it must be compulsory first to, you can say, screened out crop wise, because different kinds of crop is having different response. This work I have done in barley seed, and I find out, you can see here, 6 hour, 8 hour, 10 hours, and 12 hours. These 4 category, yeah, you can say duration of priming I have selected, and along with them, I use 4 different concentrations of zinc oxide: 25 ppm, 50 ppm, 75 ppm, and 100 ppms. And if you right side, left side is germination percent I am showing, and right side you can say electrical conductivity. I have taken the electrical conductivity of respective solutions, which I use as a seed priming at every hours. And I find out 3 things very clearly. First, duration wise, 10 hours. This 10 hours is very suitable for getting 100% germination. If you see 6 hours and 8 hours, where I have not reached up to 100% germination, even though up to time whatever I have given for the germination. But 10 hours I receive 100% germination in 2 different concentration, which is 75 and 100 ppm concentration. And once I go ahead after 10 hours, which is 12 hours, we can see over here, again, the germination percent has been declined. There is no 100% germination is there. And this thing, duration wise, 10 hours was best. And if you see the relationship of germination percent with electrical conductivity, we can observe clearly the concentration at which we received 100% germination, their EC was less. You can say minimum out of all the range. Means it has a negative relationship with germination. If EC will be less, then germination is maximum possible. So these 3 things are concentration wise, 75 and 100%, 100 ppm concentration was optimum. So similarly, I would like to suggest different like you can say pulse crop, cereal crops, and oil crops. In all these crops, people, if going to screened out accordingly, definitely he will get benefit and from the seed priming. Next. One more thing I want to show you here, chlorophyll content of the same hours of concentration and duration. Again, we can find out, we can see 10 hours is getting best for total chlorophyll content, and chlorophyll A, even though chlorophyll B was also high in the respective concentration and duration. Next. Here, in this slide, I want to discuss about the potential and benefits of seed priming. I have enlisted a few headings, even though many more headings are possible, but I use few headings. Enhanced germination performance. Once we use seed priming reagent, yeah, you can say agent, for their own purpose as per the objective set by the work, uniform and faster germination is possible. Improved seedling vigor, yes, better root and shoot development is possible. Better crop stand establishment, it reduces seedling mortality. Means if the mortality rate will reduce, definitely plant population will be better over the field. Stress tolerance, yes, it induces defense mechanism by activating different kinds of enzymes, catalase, peroxidase, superoxidase mutase, many more are possible. By using different kinds of chemicals, it is possible to get it activated. Better osmotic adjustment, yes, definitely. Osmoregulatory process is very important to get stress tolerance in the crop. Improved yield productivity. It increased the biomass and higher grain, but it is not possible directly. It is a sequential event, and one by one is possible. Cost-effective strategy. Yes, it is a very cost-effective strategy. Low input is required, and higher benefit is possible from using this priming techniques. Next. Along with the benefits, some limitations are also I observed. Limitations and challenges under this I want to share. Shorter shelf life of primed seed. This is the biggest challenge for the seed priming strategy or techniques, you can say. Once we'll prime the seed, a maximum up to 1 month we can save the seed. Later on, it will deteriorate their quality. So it need to work on this concept to increase the shelf life of primed seed if it is possible. Next one, over priming is a risk. As I discussed in my work, you can say concentration of respective chemicals and duration of the priming play a very crucial role to get it maximum benefit. So if you go for the over priming, definitely there is no benefit, and sometimes negative results are also possible. So those are working on this concept definitely have to screen out the seed as per their requirement and recommend well also. Next one is standardization issue. The same thing, once we screened out, the things will standardize, get it published, and people will follow the concept and get will benefit. Next, large scale application issue. Till now, people are using but not getting more popularity. So it is required to use it as a frontline demonstration and to make it more popular among the farmers. It is a very easy process, very cheapest process, so we'll get benefit. Next, crop and cultivar species response. As I told earlier, it is a crop, crop-wise response are very common along with the priming. So screened out as per their endospermic material content like cereals, pulses, and oil seed. These three are the very popular categories of the crop. So accordingly, duration will reduce or increase. Always may not be same. Interaction with other treatment. Yes, definitely to increase the benefit of seed priming, definitely we have to use some other, you can say, treatment also, which is possible. Like as I told you earlier, I use KSB, potassium solubilizing bacteria. In Punjab, right now I'm in Punjab, I'm working in LPU. And here, if I have gone through the literature and books which is published for the as a cultivation practices in Punjab, here potassium is not recommended because already huge amount of potassium is present in the soil. But potassium is not present in soluble form, but is present in insoluble form. So under such a condition, if you use some other treatment, like I use potassium solubilizing bacteria, so I have received very good result. Or even though I tested the concentration of transformation, you can say availability of potassium from insoluble to soluble form, I found it is very useful and it helps increase in the yield. So interaction with other, you can say, treatment is also useful to uplift the concept and benefit of seed priming. Lack of field validation. Yes, till now, as I told you, it is lacking. It is a big gap. Once the concept will popular and will get it, you can say, patent and popularized by the chemical companies to make there some formulation like other compounds are available in the market. Definitely, it will be beneficial and useful for the farmer. Next. If I talk about the future direction, as I discussed earlier, standardization of protocol is compulsory. It's very important because if you're not getting standardized or screening out properly, then benefits are not possible. Even though I personally use this concept, sometime randomly I have given more concentration, more duration, or less duration, the effect was negative. Therefore, it is essential to go at right concentration with right duration of seed priming. Crop and genotypic specific priming strategy should have to be followed by the scientific community to get it benefit. Sustainable and biodegradable agent should be used as a priming agent, which will be good for the sustainable, achieving sustainable goal also. Synergistic approach, as I discussed in the previous slide, synergistic approach is possible once will integrate the priming with other possible, you can say, combinations of the chemical. Their benefit will increase. Last one, which is very popular nowadays, AI-assisted optimization of priming protocol. I am working on this concept right now. How like other things are possible, sensor-based irrigation is possible nowadays, sensor-based many things is possible. So why this seed priming is not possible by AI-assisted tools? There is no need to go for the standardization. The system should have to already say, now its priming has been done, now you can go for the sowing. So such type of thing is possible nowadays. Once we'll integrate with the computer department, and AI assisted things is possible. Next. In the last conclusion, seed priming stand out as a practical and promising strategies to enhance the crop performance under both optimal and stressful conditions. Its potential to improve germination rate, seedling vigor, stress resilience align well with the goal of sustainable and climate-smart agriculture. It can help address both food security and climate resilient. Innovation is addressing its current limitations. So by that conclusion, I want to brief one more thing. Yes, priming is a very good strategy, but once we follow the concept, rules, regulation, as I told, the concentration and duration, I am focusing nowadays, it is very important. And without following these things, the result is not possible up to the mark. Thank you very much. Next. Yeah, thank you to all dignitaries sitting on the desk. And once again, before the end, I would like to say thanks to all the panel members who have been selected me. Believe me, and I promise all the peoples, all the committee members, I'm committed to do better for the society. Thank you to all.
Thank you, Dr. Anayatullah, for a wonderful presentation. And we wish that the young scientists who are in active research and who had been working meticulously for research in various areas, they will be instrumental in taking the society and the journal to new heights. I once again congratulate you on behalf of the society, on behalf of the president, Professor Vandana Bose, who will be concluding the session after both the lectures are there. And I wish to congratulate you and wish you a very bright future, Dr. Anayatullah. Thank you very much.
Thank you, sir.
Now I would request Dr. C. Gopalakrishnan for his lecture. Probably he will be talking on the recent advances in detection of rice diseases. And
Yes,
Dr. Gopalakrishnan, would you like
Yes.
to have the slides shared by our office, or you will share it?
Sir, I have my slides. I will try to share, sir. If there is any problem, then they can share from your
Okay,
office.
okay. Thank you very much. Please continue.
Thank you.
Thank
sir.
you.
Sir, whether slides are visible, sir?
Yeah, it is visible.
Okay, sir. Good evening, sir,
Please
and
continue.
respected chairman, Society for Plant Research, and all other members of the society. First of all, I thank all of you for giving me the opportunity and for giving this SPR award lecture. So my topic of so last time actually I have tried, but because of some technical glitches, I was not able to present. So thank you for the organizers for giving me the another opportunity. So here, these are all the some topic. Recent advances in the diagnosis of rice diseases is the topic on which I am going to speak. And I have been working on the diseases of rice for almost a decade. That's why just I have chosen this topic to talk on the rice diseases and its diagnosis. So coming to the rice diseases, like AD crops, rice also suffers from a number of plant diseases, and approximately about 15% on an average yield reduction has been reported all over the world as well as from India due to onset of different diseases. So among there are more than 50 diseases reported on rice. There are 4 or 5 important diseases. There are major one, which are responsible for considerable yield loss almost in all parts of India and of course across the globe. So among them, the first one, top priority is the rice blast. It is a fungal disease caused by Magnaborthium rice. The second one is a brown spot caused by Cocleobolus diabetes. And the third one is the sheath rot, Serocladium rice. And the fourth one is the bacterial disease, that is bacterial leaf blight of rice caused by Xanthomonas rice, Pethobor rice. And of course, the emerging one. So before 10 years, it was considered as a minor. Now it is an emerging, that is the rice fall smut. Again, another fungal disease caused by Aspergillosis virus. So the area of my current area work is the development of a diagnostic kit for the early detection of seed-borne pathogens of rice. Because most of the fungal diseases in rice, they are all seed-borne. They are all seed transmitted. So it is very crucial to detect the health of the seed before importing or exporting a seed. So we should be in a position to detect the presence of important seed-borne pathogens on a real-time basis. So as we know, most of the techniques now, what we rely on is standard blotter technique. So basically, it is an incubation method. It takes about 8 days of incubation, and requires a lot of time. In the microscope, and a lot of technical expertise is required. Again, there are overlapping in the identification of the seed-borne pathogens. So taking in all those things into consideration. So if there is a diagnostic kit, that can detect the pathogens immediately on a real-time basis, that would be very useful in the context of quarantine as well as seed certification, as well as for the other seed health in general. So keeping in this mind, I have developed a lamp-based technique. So it is a thermal amplification assay. So it is also nucleic acid based test or assay similar to PCR, but it actually a very simple method when compared to PCR. So it can be performed even under resource power settings without the requirement for a PCR machine or a thermocycler. So presently I'm working on this LAMP technology. So we have developed the diagnostic assay, LAMP assay for these 4 pathogens. The first pathogen we have started was the blast, number 1 disease in rice. So as you know that this blast actually, it infects almost all parts of rice. So it infects the earhead. So that we call it as a neck blast. So that is a very serious one. So if there is a neck blast infection, so all the photosynthetes which are produced in the leaves may not be transferred into the grains, resulting in the chaffy grains. So such a very serious pathogen. So we have developed a technique, LAMP-based assay for the detection of these Magnaborthium rice in rice seeds. It's a seed-borne pathogen. So coming to the loop mediated isothermal amplification assay, or in short LAMP assay. So these are all the important advantages of these lamp assay when we are comparing to other assay, especially when compared to PCR. So this lamp, it has the ability to amplify the nucleic acids under isothermal conditions in the range of 60 to 65 degrees Celsius. So there is no need for 3 different temperature regimes, as in the case of PCR. So all the amplification, it happens only in a single temperature. So that, of course, we have to standardize for each pathogens. And it is a cost-effective reaction equipment. There is no need for the costly equipment. And when compared to PCR, the amplification efficiency is also more. So the DNA is being amplified 10 to the power of 9 to 10 to the power of 10 times. So within a very short period of 30 to 60 minutes. So we can get the results within 30 minutes. And both amplification as well as detection occurs at one step, the same steps simultaneously it occurs, and it helps in the early detection of the plant pathogens. So these are all the advantages of the lamp assay in general. So as I said, this for blast that is caused by Magnoporthi oryzae. So this Magnoporthi oryzae, it can be detected using this lamp assay. So it can be using a small quantity of the pure DNA sample. So it can be, this assay can be conducted in a 25 microliter tube, PCR tube. So actually this lamp reaction mixer typically consists of 3 sets of primer. So forward primers, there are 2 forward primers, inner primer and outer primer, then 2 backward primers, outer primers, backward outer primer and backward inner primer, and there are 2 blue primers, forward and backward primers. So apart from the DNA, betaine solution, PST DNA polymerase, magnesium sulfate, thermal reaction buffer, then hydroxynaphtol blue dye, so which actually helps in the identification of the positive and negative samples by an naked eye. Then these are all the reaction mixtures. So all these reaction mixtures, it has to be standardized for each pathogen. And apart from these reaction mixtures, the reaction time and reaction temperature has also been standardized for each pathogen. So this lamp assay can be shared either by keeping mixtures in the PCR machine or in any simple heating block. Because PCR is there, we can well and good. But when the machine is not available, even in a heating, using a simple heating block like a water bath with the temperature control, we can perform this lamp assay. So what we are seeing is the result of this lamp assay. So there are 7 or 8 PCR tubes you can see. These are all the each samples. The first 1 to 7, they contain Magnoporthi oryzae, template DNA of Magnoporthi oryzae. The 8th one is the negative control. That is the only, it contains nucleus-free matter. There is no DNA of the Magnoporthi oryzae. So at the end of the 60 minutes after lamp reaction, you can see the development of sky blue color development in the first 7 tubes. So it indicates the positive reaction. That means all these 7 samples, they contain Magnoporthi oryzae. The 8th one is the negative control, so it remains as violet. So we can visibly see the results within 30 minutes. So that is the advantage of this one. And again, if you want to run this on this amplification, if you run it on agarose gel 1% agarose gel electrophoresis, you can see the ladder-like pattern of the amplification in the positive samples in all the 7 samples, while in the negative there is no amplification. So in both ways we can confirm whether the reaction is positive or negative. So this is how we have did using the 7 isolates of the Magnoporthi oryzae. And the next step is now we have to check whether these whatever the lamp assay we have developed is very specific to rice blast pathogen, Magnoporthi oryzae only, or whether there is any cross reaction takes place with other pathogens. So for that we have taken 7 other important pathogens. There's Helminthosporium oryzae, Plasmapora viticola, then Erysifi, and Fusarium oxysporum cubens, which causes banana wilt, then Critotrichum capsicum, then Sclerospora sorghii, and Sclerospora graminicola. These are all the downy mildew pathogens of maize and pearl millet. So we have tested for this cross reactivity of the lamp assay, and we can see only in the first one, so which contains the culture DNA of the Magnoporthi oryzae, there was a positive reaction. There's a development of a sky blue color. In remaining all other things, including the negative controlled, and other pathogens, there was no reaction, lamp reaction. So it ensures that whatever the primers which we have developed is very specific to Magnaportio rice. So apart from that specificity, we have to assess the sensitivity of this lamp assay also. So we have tested this sensitivity by diluting the DNA of this Magnaportio rice to up to 1 picogram level. So that line 1 is the 100 nanogram, then 10 nanogram, 1 nanogram. So likewise serially diluted. So up to 1 picogram, we have diluted it. So this lamp assay, you can see it can amplify even a 10 picogram of DNA in the sample. So very sensitive one. Of course, below 10 picogram, it cannot amplify it. So you can see the reactions remains negative in the 9 and 10. So the same thing was confirmed on the by seeing the lamp products in the 1% agarose-gel apophoresis. So that is how we have standardized the sensitivity, validated its specificity. So this is with regard to blast. So likewise, the second pathogen that is known as the brown spot of rice, that is caused by Coccolobulus miavenus or bipolaris oryzae, that is the asexual stage. Here also we have developed a lamp assay. So the lamp reagents basically is similar to what I have described for the blast pathogen Magnaportio oryzae. Of course, the components, the reagents, the ratio that actually I have standardized. This is the specific. For any pathogen, when we are going for a lamp assay, the designing primer, that is the most important, most crucial factor. So here a set of 6 lamp primers were designed by targeting the 2 of the glycosyl-transferase protein. So it is within a conserved region, very specific to bipolaris oryzae or Coccolobulus miavenus. So using the Primer Explorer software version 5, and we have tested again for its specificity and sensitivity, and found that an incubation temperature of 60 degrees Celsius for 30 minutes was found to be optimum for the detection of bipolaris oryzae. So it was very sensitive. It was up to 10 okay. So we edited this developed lamp assay using the 35 different isolates collected from different locations of Tamil Nadu. And the lamp assay was tested for the detection of bipolaris oryzae artificial inoculated seed sample. We have also validated it by testing the presence of these bipolaris oryzae on rice leaf as well as seed samples. So these are all the quantity of different 35 isolates of bipolaris oryzae we have isolated. So these are all the cultures of some of the isolates of bipolaris oryzae. So this is the in silico hybridization of the lamp primers. So we have designed the primers for the bipolaris oryzae. We have targeted the exon 2 of the glycosylated transferase family 19 protein. So initially we have characterized the bipolaris oryzae as a pathologist using the ITS1 and ITS4 primers. And these are all the conditions we have standardized for the detection of the bipolaris oryzae. These are all the lab reagents, and this is the incubation temperature, that is 60 degrees Celsius for 30 minutes. The third pathogen we have tried with was Serocladium oryzae, actually which causes sheath rot disease in rice. So which actually this disease manifests typically during the emergence of the panicles. So the boot leaf sheath is infected by the Serocladium oryzae, resulting in the earhead choking. So the earhead may not come out effectively, maybe a partial or total choking earheads. So we have isolated 59 isolates, and we have sequenced it, and we have developed again 3 sets of lamp primers. And these are all the different cultures. Some of the cultures are isolates of Serocladium oryzae we have isolated from the sheath rot infected rice plants. And we have characterized or identified using the ITS1, ITS4 primers. And again, coming to the designing of the lamp primers, in the case of Serocladium oryzae, we have targeted the immunoglobulin gene and designed 6 primers. So these are all the sequences of the 6 primers. So these are all the forward or backward or inner primer and loop forward and loop backward primers. These are the diameters of the length of the different primers. And we have tried for the developing a lamp assay for the bacterial leaf blight caused by Xanthomonas varaceae, Pethovaraceae. Of course, we have targeted hypothetical protein, and the work is still going on. We have not yet completed this work, but we are in the process of designing the lamp primer for the Xanthomonas varaceae, Pethovaraceae also. So these are all some of the isolates we have isolated of isolates of Xanthomonas varaceae, Pethovaraceae, which causes bacterial leaf blight rice. So this is the in silico hybridization for lamp primers targeting the beta tubulin gene of the Serrapillidium varaceae. So we have validated the developed lamp assay Serrapillidium varaceae using different isolates of the Serrapillidium varaceae. So all of Serrapillidium varaceae, you can see the amplification all the 7 isolates of Serrapillidium varaceae. So So these are all the conditions or the of the lamp assay for the detection of Serrapillidium. So we have tested the presence of Serrapillidium varaceae in an artificially inoculated seed samples, and it showed positive reaction only in the inoculated samples. The lamp assay was validated from the rice leaf as well as the seed samples collected from the different locations. So these are all the lamp assays which were validated using different isolates of the Serrapillidium varaceae. Totally there were 34 isolates we have tested, and it actually amplified all the isolates. So the optimal concentration of the lamp reagents like magnesium sulfate, betaine, DNT base, BSD DNA polymerase, and HNB dye in lamp assay was standardized. The incubation temperature of 65 degrees Celsius and for a duration of 30 minutes was found to be ideal for the detection of SO rice from rice seeds as well as infected plants. So after developing all these lamp assays, and we are also in the process of developing a lamp assay for the Paulsman pathogen and emerging disease of rice. So that and bacterial leaf blight pathogen, it is still in progress. And another area where we are working is the reaction diagnostic kit. So what we are all doing is inside the lab in a test tube. But if you are able to do it on a simple membrane-based kit, we can a portable kit. So all the tests can be performed in the real field or on site, so that we can make a decision on a real-time basis. So that is how we are trying. So just we have started with 4 different types of membrane. The first one is the Wattman #1 chromatography grade paper. The second one is polyether sulfone membrane, and the third one is glass fiber. The fourth one is cellulose acetate, and then last one is nitrocellulose membrane. So we have tried different membranes, and we have dried all these lamp reagents in ideal conditions and stored in the refrigerator conditions. And we have used details of the kit, but we were able to get some of the results in the colorimetric assay. However, when we were going for, so in a membrane-based test, so there was no significant difference between the positive and the negative. So almost both are look similar. Only the negative sample is somewhat pale, the positive sample is somewhat dark black. That also when we are looking under specific UV light, not under normal naked eye. So of course, with the use of HNB dye, we can able to see the results, positive and negative results. But yes, on the membrane, we were not able to do it. So we are just working on it. We are just trying different types of membranes and different types of the lamp assay mixers also, to which can work better on a membrane-based system. So these are all the some of the area where we are working on it. And these are all some of the publications we have made in 2025 up to this June. And I am working in the All India Coordinated Research Project on rice as a pathologist. So last year we have released 2 varieties for Tamil Nadu, O58, that Coimbatore 58, and Coimbatore rice hybrid 5, one variety and one hybrid for cultivation in Tamil Nadu. So these are all the important characters of these 2 rice varieties, which are moderately resistant to blast, rice thumbro and brown spot, and CoRH5 with moderate resistance to leaf blast, nick blast, and grain discoloration. So again, with this, I thank again the organizers for giving me this opportunity. Again, I thank you all. Thank you, sir.
Thank you very much, Dr. Gopalakrishnan, for wonderful presentation. And of course, these award lectures are not only meant for the conferment of award, but our fellow scientists and the society members, they at least know that in what field our esteemed awardees they are working. Sometimes they need to collaborate with each other, so they get an opportunity to collaborate with them. Sometimes they need to seek some expertise. They also may contact you sometimes. So this is a wonderful platform when we meet together. So we discuss so many things and interact with each other. Vegetos is now having 98 editors from 47 countries, as you all know. And I would request all our esteemed awardees to keep on visiting our website, www.vegetosindia.org, for all updates, whatever are there, and keep motivating, because you are the ambassador. You are the ambassador of the society. So ultimately, the society expands through its members and through the awardees. So ultimately, they just motivate the scientists, young researchers, and fellow colleagues to contribute good research to the journal. One very important fatal trend is prevailing nowadays, that with the help of ChatGPT and artificial intelligence, very young guest faculties or those who are in the beginning of their career, they are writing review articles. This is fatal for the cause of science, which I understand. Because unless and until a person has established himself in a particular research area, he is not able to write a review article. If I, after 41 years of my service, if I write a review article on the topic of my expertise, that speaks sound. If Professor Bandna Bose, she writes some very good review article on the field of her expertise after 40 years of her service, then it sounds good. But if a person who has just joined and he is not attaining the permanent position even, they have started writing review articles. It could have been possible only because of ChatGPT and artificial intelligence. They have not to exert anything. Just give a command to artificial intelligence and the entire paper will be ready. So Vegetos and Springer Nature has developed a tool which can identify whether this paper has been written by taking the help of AI or it is the original research. Because we have kept so many parameters and new parameters have been inducted, with the help of which we are trying to make our journal absolutely transparent. There can be no dirty practices, no fake practices, nothing can be done. So we are monitoring each and every article at every step before sending it to the publication. So with these few words, I would like to request Professor Banda Bose, who had been not keeping good health for few days or few months, but even then, I am grateful to her because she spare a lot of time for academics. This is the passion of entire group of Society for Plant Research, which we had been working for it. So I would request Professor Banda Bose to kindly conclude and give blessings to the esteemed awardees. Professor Banda Bose.
Yes. I am audible, sir? I am audible? Yeah.
Yeah, you are audible.
First of all, I pay my respect to Professor S.K. Bhatnagar, the founder chairman of SPR. This SPR society is running since 1988, and it has covered 36 years or more than 36 years of its journey and publishing a very, very good journal, Visitors from Springer Nature. As a person, Professor Bhatnagar, taking very much pain about the things related to the academics. I am very much respectful organizing such a webinar for the fellow awardees for 3 days. Today, 2 lectures were left, and those again he has arranged for the awardees, and Dr. Anayatullah Siddiqui, he has given a very good lecture regarding the seed priming technology and its benefits and challenges also. The C. Gopal Krishna, Dr. C. Gopal Krishna has given a very good lecture about the recent advancement in diagnosis of rice diseases, and he fungal diseases, those are seed-borne, by elaborating the LAMP technique, by LAMP technique essay, how helps in the early detection of the disease. Both the paper has immense importance in the field of plant science. I personally feel very much delighted to be a part of all the webinar of SPR Fellow Awardees, which has occurred which has covered all the portions of plant sciences. Really, it's a very good opportunity for me to attend this on time. Now we are in the era of gene editing, and nowadays in case of plants, crops, and also human diseases. So we have to go ahead by a quick march, and that is possible with young, enthusiastic, and dedicated scientists like you people. I congratulate all the fellow awardees 2025 of SPR and request all of you to participate all the events and works of SPR for its all-time growth. And I also pay my respect to all dignitaries who are presented in this meeting. And lastly, I pay my thanks to Professor Bhatnagar for organizing such a good meeting. Thank you. Thank you to all of you.
Thank you very much, Professor Banda Bose, for a very encouraging remark. And I wish that if our esteemed young fellow awardees, they are getting blessings from such a senior scientist, they should feel encouraged. And getting fellow award is the start of this type of journey, and I hope that they will have more crowns to their more feathers to their crown, and ultimately they will keep on progressing and contributing good science, good research, and will help the society to grow. Thank you all very much.
Thank
And the awards shall be dispatched within this week by our office, and kindly keep contacting our office for that. Thank you all. Thank you, Professor Bandana Bose, and all our esteemed